it is on presentation. Yeah, thank you.
Yeah, awesome. So let me check with you because so on the side of my screen I can see you know, all these windows like with you.
We can say that.
Okay, maybe I can just make it small so I can see someone. Okay.
Okay.
Cool.
So let's get started. And hi everyone, welcome to the Nanomotor Media Talk. It's my great pleasure to introduce our today's speaker, Dr. Stefania Ketzetzi. She comes from Leiden University, and her talk will be on the Wall effects on the motion of synthetic microswimmers. So let's welcome Stefania to share research. So please go ahead, Stefania.
Yeah, thank you very much. Thank you for the introduction, and thank you also for the invitation. It's I'm very excited to be here today. So hi everybody, my name is Stefania. So for the past 4 years, I have been a PhD student at Leiden University in the Netherlands. And as you can see from the background, so during my PhD research, I have been working with colloidal swimmers, and specifically with catalytic colloidal swimmers. And in particular, I have been interested in understanding how walls affect the motion of these swimmers. And so in today's talk, I will tell you about recent work that we've published actually at the end of 2020. And in this work, we propose a new method that one can use to measure the distance between a swimmer and a wall. And this method is a diffusion-based height method. And before I proceed, I want to thank and to acknowledge my supervisor, Daniela Kraft, and also our collaborator, Kraft from Utrecht University, who has been working on the modeling side of our project and has been helping with interpreting our experiments. So what should you expect from this talk? I'm going to start by motivating why walls are important. And I'm also going to tell you a bit about the research that has led us to conduct the work that is presented in this publication. And then, of course, I'm going to introduce our method for measuring the distance, as well as tell you a bit about what kind of insights we have obtained when we applied this method to catalytic swimmers. So I want to start by showing you this movie because I think it's an excellent example of how complex motion in the microscale can be. And you may have seen this movie already, but in case you have not, for this experiment, a blood film was adhered on a glass surface. And this movie follows the motion of this neutrophil. So it's a white blood cell whose mission is to basically find and to eliminate these tumble-shaped particles, which are actually bacteria. So to do that, this neutrophil has to constantly adjust and adapt its shape and its motion to the environment around it, which also is changing continuously. And as you can see, this is a movie that has been taken several decades ago, but already this indicates that researchers have long been interested in understanding the complex motion that microswimmers perform inside complex environments. So fast forward to today, we know that biological swimmers have affinity for walls. For example, we know that bacteria form biofilms on surfaces which facilitate their spreading and their cooperation and also the capture of nutrients. So biological swimmers have affinity for walls, and then in turn, walls also influence their motion. For example, by causing accumulation and retention on their sides, but also it's known that walls can significantly even modify the trajectories of the swimmers. For example, bacteria like E. coli, when they move in the bulk, they typically perform a run and tumble type of motion. However, when they move near a wall, they swim in circles. And in fact, the boundary condition on the wall influences the direction of motion. So as we know also from this series, nowadays a lot of research also focuses on preparing synthetic swimmers, which initially they were supposed to mimic the motion of the biological swimmers. And in fact, as I hope you can see from this video side by side, these movies show that we are already in the right direction in preparing biomimetic systems. So these synthetic systems are very useful for us, not only because we can use them as model systems to understand out of equilibrium behavior and processes in active matter, but also because they are prominent candidates for applications, for example, in biomedicine or environmental remediation, where for example, the swimmers could transport and deliver cargo in the microscale. And of course, for applications, we would need to have autonomous particles that are also able to adjust and to adapt their motion to their environment, much like the neutrophil that I showed you at the beginning. So a popular model system, especially when it comes to the autonomy, is the so-called catalytic microswimmers. And as I said, these are also the particles that we have been studying in our work. So what you see here is a spherical colloid that is half coated with a platinum layer and is also suspended in a solution that contains a part of hydrogen peroxide, in which it, of course, exhibits the autonomous motion that you see, directed motion that you see here. So this is seemingly a simple system, but as we very quickly came to realize, there actually it's not as simple as one may think. So there are actually open questions pertaining to it. And probably the most important one goes back to the propulsion mechanism for these particles itself. So the general consensus nowadays is that these particles move through a type called that resembles self-phoresis. However, the exact details, as I will come back later, of the mechanism are still missing. Now, catalytic swimmers, it's something that is very noticeable about them is that they also have strong affinity for walls, much like I already discussed earlier for biological swimmers. And this affinity is something that researchers have already noticed, and they have already been using as a means to guide the motion of these particles. And going back to the video that I showed you earlier, I will just now show you a few snapshots. So what we see, and I didn't mention earlier, is that, of course, the motion that the particle performs is motion in 2 dimensions. And what I didn't say is that this motion happens on top of the wall. So basically, we add the solution that contains the particles inside the container. And what these particles do, sooner or later, they will find the boundary of the container, in this case, the bottom wall, and then they swim there, they self-propel there, and remain there, moving in 2 dimensions above the wall. So since the motion happens near the wall, and since we measure it there, the question that we initially had was, are there any effects on the motion of the particles coming from the wall? And we hope that this may help us in understanding a bit better the propulsion mechanism. So just to briefly show you this, because it's something that we did already in our previous work. So in this case, we took the same particles, we fixed the experimental conditions, and then we only changed the wall in the experiment. And indeed, we found that the wall affects the speed of the particles. And not only us, but also actually many other groups have already been reporting in the literature wall effects coming onto the speed of the swimmers. So this brings me to the question of today's talk. So since we and others are being able to measure wall effects, this suggests that the particles should move sufficiently close to the wall. But the question is how close? So the best estimate to date we have from the experiments by Sinchen, in which they used steps that they printed on top of their walls. And what they found was that sometimes the particles were able to cross over these steps, and sometimes they would just be stopped. And this depended on the height of the steps. So this is already sort of can serve as a bound for the distance between a particle and a wall. So it gives us some sort of expectation when it comes to the distance. But still the question remains, how can we measure this distance? So just to say that in general, it may come to mind that there are a few methods already in the literature that one can use to measure the distance between a particle and a wall. For example, total internal reflection microscopy or holographic microscopy have already been used. However, I just want to note that in our case, we have been working, as I said, with particles that are equipped with a metal on one side. And this metal actually makes it hard for us to use the methods that I mentioned before. So what we wanted to do in this work was to find a method that is quite general so that we could use it for any sphere, even the ones that have a platinum coating, and also that it would be quite simple. And so what we thought to do was our method basically relies on the hydrodynamic mobility of the particles and the fact that this is reduced by the presence of the wall. So as a first step in our method, what we do is we extract the diffusion coefficient of the particle, which we can do from the mean square displacement that we can calculate from the particle positions in time. And basically from the mean square displacement, for example, in the case of a Brownian particle, we can fit it with the Einstein relation and extract this way the diffusion coefficient. So as a second step in our method, we also need to calculate using the Einstein-Stokes equation, the diffusion coefficient of the particle in bulk. Why do we do that? Because as the next step, what we are going to do is that we want to relate the reduction or the ratio of the diffusion coefficients to a height from the wall. And this luckily has already been, there are already predictions existing in the literature. For example, when we are in the lubrication regime, so very close to the wall, this paper by Goldman, Cox, and Brenner shows that we can relate the reduction of the diffusion coefficient to a height through this mathematical expression, as well as when we are far from the wall, we could use a Paxton equation to do so, which actually Paxton equation has also been proven in the literature already in experiments. So when we did that, in our case, we found that this ratio of diffusion coefficients fell within this rectangular area. So that means that neither of the two expressions was accurate enough. So both expressions would give an error of about 20%. So one of them would underestimate and the other one would overestimate the height. So what our collaborator did is that he used numerical data that exists in the literature, and then he performed a third order interpolation of the log-log variant of this combined data. And basically he came up with this prediction here, this dashed line. I hope you can see it in this graph. So basically now we have a prediction that relates the reduction of the diffusion coefficient to a height from the wall for the entire separation distance regime. So of course we want to apply this method to platinum coated particles. In this case, of course, when they're in the active state, so in the hydrogen peroxide, in this case, we cannot use the MSD expression that I showed you before, so the Einstein relation. But luckily for us, also, there is a lot of literature that discusses the mean square displacement for the case of an active particle. And I refer you to these articles here. So in these articles, you can also find the full mean square displacement expression for both the short-term regime and also the long-term regime. Here, I'm only showing you the short-term regime. And basically, this is what the mean square displacement expression reduces to. So in this regime, if we are able to capture it, we can fit the mean square displacement data, and we can decompose basically the two contributions to the mean square displacement. One comes from this passive component that contains the diffusion coefficient, which is of interest to us, but also the active component that contains the speed of the particle. So by fitting this mean square displacement data in the short-term regime, we can obtain the diffusion coefficient also in the case of an active particle. So going back to our method, what we do is we measure the mean square displacement of an active particle. We extract the diffusion coefficient from the measurement, as I described now, and then we use the prediction from the previous slide, and this gives us the height the height of the active particle from the wall in the active state. So that was already quite cool because it was the first time that we could measure and have a direct indication of what is the distance between an active particle and the wall. So now that we have this method, what can we do? We were interested to see how the distance between a particle and the wall changes when we change parameters in the system. And the parameters that we looked at are the ones that we already know from the that are important in the case of passive particles. For example, in the case of passive particles, we know that if we add salt in the solution, because salt would screen out the charges, the particle would move closer to the wall. So we expect that the distance in a passive system would decrease with the salt, with an increase in salt concentration. So when we did this experiment in the case of an active particle, we surprisingly found that the distance was actually remaining constant. And in fact, the distance that came out of these measurements of the order of 250 nanometers is actually what is expected based on, for example, the experiments by Simchen that I mentioned earlier. Then we were interested in seeing how the surface charge of the particle affects the distance. So we performed experiments in which we used particles made of the same material and the same size and platinum coating. So basically the buoyant component in this case is the same in all cases. And what we did, we changed the surface charge of the particle, which we can quantify through the zeta potential of the particle surface. And we did that by changing the functionalization of the particles. So we went from particles that were negatively charged to particles that were positively charged. And in this case, also surprisingly, we found that the distance remained constant. This is contrary to what we expect in the case of the passive system, because in the passive system, when we are measuring above, for example, a glass wall, we expect that the particles that are positively charged would be stuck on the wall. But this is not the case in the active particle state. So, and then again, with the constant distance that we find is similar to the one that we saw in the salt case. Now, as a next step, we also wanted to see how the radius of the particle would affect the height from the wall. And what we did here is we took particles made of the same material, so the same surface functionalization, so the zeta potentials of these particles were similar. And what we did is that we changed the size of the particles. And then again, also surprisingly, we found that the distance between the particles and the walls in this case was also constant and similar within error to what we have measured before. So this led us to this behavior, this tendency of the particles to move at fixed height from the walls with these changes in also irrespective of these parameters in the system. We coined it ipsotaxis. What we find essentially is that our catalytic particles swim at a height of a few hundred, of the order of a few hundred nanometers from the wall, and this height is actually quite robust. So then the question is, because from these experiments, it's surprising that electrostatics in this case and also buoyancy are not the primary contributors to this tendency that we find. So our hypothesis was then that this tendency is something that happens because of the activity of the particles. And to test for this hypothesis, we performed experiments in which we start from the passive state, so there is no fuel, no hydrogen peroxide, and we measure just by adding at first a bit of hydrogen peroxide. We immediately saw that the particles would again move from further from the wall. They would just move, they would come closer to the wall, and then very fast this the distance from the wall would plateau. And this also coincided with that in the same range of hydrogen peroxide, and already from these similarly low hydrogen peroxide concentrations, the speed of the particles also plateau. So this actually verified our hypothesis that the ipsotactic tendency that we measure is induced by the activity. And when I say induced by the activity, I mean that we hypothesize that this happens because of the phoretic and osmotic flows that surround the particles and the wall. So now the question is whether these experiments can give us, what can they tell us, if anything, about in relation to the propulsion mechanism. So as I said at the beginning, the propulsion mechanism for these particles is actually debated. Initially, self-diffuse geophoresis was proposed to describe their motion. However, because of ionic effects, researchers proposed that self-electrophoresis may play a role here, and actually it could even be, so current thinking favors that self-electrophoresis is the propulsion mechanism for these particles. Now, what can our experiments tell us? So going back to the experiments where we change the zeta potential of the particles, I didn't show you before this, but what we find when it comes to the speed of the particles is that it remains not only, in this case, not only the height remains constant, but also the speed of the particles remains constant. So the swimmer surface charge or the particle surface charge does not affect the speed. However, going back to self-electrophoresis or also ionic diffusion-phoresis, this is not what is predicted. So for example, self-electrophoresis proposes that the speed should scale with the zeta potential. And this is something that we do not see here, and it led us to think that it could be that ionic effects are not important around the swimmer itself. However, going back to our findings, the speed findings that we have when we add salt in the solution, we find that here it seems that ionic effects are actually important. And the reason is that when we add salt in the solution, much like other researchers have seen before, the speed of the particle decreases. Now, we can capture this decreasing trend in our data by feeding it with this expression that I saw here on the right. And so this is work done by our collaborator. So what we propose is that based on Anderson and the types of self-heretic mechanism described in this famous article, only ionic diffusion scales in this way. So if we combine this with our previous finding that I showed you in the previous slide, how we interpret this expression that fits our data is that we propose that the first term is a term that represents the non-ionic effects of the particle, and that however, the second term includes the ionic effects that happen on the wall. So to summarize this, what we think is happening is that that there is a fluid flow that is created on top of the wall because of this salt gradient, so this ionic gradient on the wall, and this fluid flow opposes the self-propulsion, and it's what causes the particles to slow down. So if you would like to, also in view of time, if you would like to know more, again, I refer you to our article. So to summarize what I've told you is that we find that the distance between a particle and a wall can be measured from from the diffusive component of the motion. And we also find that the particles, the catalytic particles that we use in our experiment move at a few hundred nanometers from the wall. And we also find that this height is actually quite robust. And what we proposed in our work is that it seems that walls are actually important, and we propose that to understand the propulsion mechanism in future modelings, we should consider also the fact that the particles move very close to the wall and also incorporate the effect of the wall. And before I conclude this talk, I want to give you a bit of future direction. So as I said at the beginning, a great motivation comes from biological swimmers moving in complex environments. So as a next step, what we did is we wanted to also increase the complexity in our environment. So we performed the experiments in which we 3D printed obstacles on the wall. And in this way, we managed to go from a two-dimensional motion to actually confine motion in one dimension. And we find some really interesting behaviors when it comes to the speed of the particles, as well as we also find that particles can assemble into self-propelled chains. And if you would like to know more, please look at this article that we uploaded on arXiv. And with that, I want to, of course, thank again Daniela, my supervisor, and also Joost, our collaborator, and of course, you for the attention. Thank you very much.
Okay, thank you, Stefania. Thank you for your nice talk. Now we are going to the Q&A session, and please feel free to unmute yourself and ask a question.
Hi, Stefania.
Hello.
Can you hear me?
Yes.
Thank you for your nice talk. I have two small questions.
Yeah.
So the first one is when you talk about the diffusion, you also cite diffusion of the bug. So I don't know what the bug mean. How do you measure this? Like you calculate, yeah.
Yes, you can calculate through the Einstein-Stokes equation. It basically, yeah, I could send you something if you want, but it's basically the thermal energy versus the, so it's the kBT versus 6 pi times the viscosity times the radius of the particle. And with this expression, you can calculate what the diffusion coefficient of the particle of the same radius would be if the particle would move further from the wall.
Okay, so I mean it's not an active particle, or it's just passive particle, just not active.
Yes, exactly. So the idea is that you would like to, in general, the idea of this method is that you would like to extract the passive component. Even in the case of an active particle, you want to extract the passive component of the motion of the active particle.
Ah, okay. Okay. So now, yeah. Thank you. And another one is when you discuss about how the surface charge influence the height, you also change from negative charge to positive charge. You also said like for passive particles, it might stuck on the slides, but for active one, it doesn't. So
yes.
It's really interesting for me. Like, yeah, could you explain or give an idea why it's happened for active, not stuck?
Yes, exactly. So, so what we propose is that so to understand this, we should think about what is actually what causes the propulsion of the particle. And the current idea that we have is that there are these flows that are created along the particle surface. This is these are the phoretic flows that also factor into the fluid, the speed of your fluid. But also what we saw in our work and also in our previous work is that the fluid flows along the wall can also be important. So around the particle, so in this particle wall system, you have these fluid phoretic flows and osmotic flows. And we think that these are responsible for the fact that the particle is sort of kept or sort of balanced at some this is basically what sort of lifts the particle and keeps it at a fixed distance from the wall.
Okay.
So we basically what we say is that the electrostatic contribution is not important or is not a dominant contribution to this ipsotactic, how we call it, tendency that we observe in the experiment.
Okay, so yeah, you mean the electrostatic induction is not strong compared to the flow it induced. Okay, for this case, yeah. Thank you.
Thank you.
So any more questions from the audience? Stefania, I have a question. So what do you think if the microswimmers is other type of microswimmer, like the biomatic rods? Did you perform the experiments with this type of microswimmers? Is that fit your theory too?
So we have never done experiments with other kinds of swimmers. Which theory do you mean? You mean when it comes to the propulsion, or the fact, or when it comes to whether we expect that these particles will also have an ipsotactic behavior? I didn't understand what the question.
I mean the height between the particles and the slide.
I think so. Yes, I expect them to be moving at just because I think in my mind now I have some experiments. For example, I don't remember exactly which paper it was, but I have seen videos of rod-like particles moving, and they also perform, from what I remember, two-dimensional motion. And if I think of in the videos, for example, you could see that they also propel roughly along the same plane above the wall. But this is just what I remember. Please correct me if I'm wrong. You probably have a lot of experience with rod-like electrophoretic particles like that. Is that the case? Is it how they also move in 2 dimensions above the wall?
Yeah.
Okay. Yeah, I think, I don't know if you could use our prediction to measure the height, because I suppose it holds for spherical particles, but it would be interesting to see what would be the height from the wall for these particles. I don't have an answer to this because I've never worked with particles like that.
Okay, okay. Thank you. Thank you. Thank you for your answer.
Thank you. But now, if I think I remember an article, a Physical Review Letters article from, it could be 10 years ago. I think they mentioned somewhere that they expect the distance for electrophoretic rods to be of the order of tens of nanometers, actually. But I don't know if they have, I don't think that they have ever measured it. It's probably an expectation based on what they see from their microscopy images. Yeah, I can send you this article if you would like. I think I can find it.
Yeah, yeah, thank you.
Hello.
Hi.
Hi, Stephanie. Okay, thanks for your nice talk. So I have one more questions about this. So your particles, the mechanism is based on the self-diffusion process, right? You mean?
Well, okay. We do not know. It's probably the best answer to this. As I said, the current thinking favors, because of these ionic effects that people and we also have seen that are important for these particles, probably the favor is towards self-electrophoresis. What we said in our work is that it could be that the propulsion has still the form of an ionic self-diffusiophoresis or diffusiosmosis, actually. Also, if you take into account the fact that the flows that are created along the wall can also be important and can also contribute to the motion. So it could be a good way to check for this. It could be, for example, to take the particles, because we have never done that. We've never done three-dimensional motion, three-dimensional measurements for our particles. It could be a good check to lift them far away from the wall and see what happens to their motion, if it decreases and how it decreases when you add this salt in solution. Because what we propose is that these salt gradients could be important because of the wall and how they act on the wall. But we actually have, yeah, I think to do 3D measurements would be a next good step to understand better what is happening. Just what our work sort of put forward is that it could be that the main contribution is still diffusiophoretic if the wall can sort of cover for this reduction that we see in the speed.
Okay.
But
I see.
Yeah, it's of course it's an open question.
So what do you mean about the flow on the surface? You mean the surface is the surface of the substrate or surface of the particles?
Yeah, exactly. So, so the initial picture, also what I showed at the very beginning when I first discussed the motion of these particles in the hydrogen peroxide, we think that this, basically, generally the idea so far was that these particles, they have a fluid flow that is created around them, around the surface of the particles.
Yes, yeah.
But this is more of a bulk sort of I mean, of course it's true, but it's also a bit of an image of expectation of how would the particle move away from the wall. Now, if you have them near a wall, and in the same way that these solute molecules create this fluid flow along the particle, they can also create a fluid flow along the wall. So you have a fluid flow, an osmotic flow along the wall, and a phoretic flow along the particle. But when you're moving so close to the wall, it could be that this osmotic flow on the wall couples back to your particle, and it modifies the speed of your particle.
Okay. Okay. So, so you mean this flow get rid of the particles stuck on the surface when we change the surface charge, right? It's it. So because of this
Sorry,
surface.
I didn't get that. Can you explain again?
You mean this flow? You mean that you change the surface charge of the particles, and for the active particles that didn't stuck on the surface, it was because of this flow on the near the surface get rid of the particles stuck on the surface.
Oh,
it's either right.
Well, I don't know if I would say it like this, but indeed, what we think is happening is that this particle is kept at this distance because this is something that you do not see in the case of the passive particles, right? So in the case when there is no activity, so there is no phoretic flow, there is no osmotic flow, because if you don't have activity, like activity in the sense that you don't have a fuel that you decompose, so you don't have this asymmetric distribution of solute molecules, so you don't have this kind of flows that are created. So in this case, you don't see this behavior. So, and from what we have seen, again, as I said also earlier, the electrostatics, in the sense that the zeta potential was not important in the experiment, or the buoyant component was not important in the experiment, this led us to just hypothesize, and we also seem to recover this from the additional measurements that we performed, that this is something that happens because it is activity induced. It's a phenomenon that is activity induced. So you need to have these flows that you have in the active state to see this sort of a constant height tendency of the particles.
Okay. Okay. See. Okay. Thank you.
Can I have a small question based on this? So I'm curious about when you change the surface charge, if the motion direction also changed? Because
no, actually it does not. No, we didn't see that. Also, the particles that were positively charged, they also moved away from the platinum, and so we did not see the reversal in the motion. So again, this we thought speaks more in favor of a wall effect rather than an effect that happens around the particle. Let's say, I don't know if that was very clear.
Okay. So maybe the electric thing is really less there, maybe. And also, a small question is, when you add a lot of salts, the salt concentration when it's high, did you see the particle try to stuck on the slides also?
That's a good question. Yes, eventually. I think the reason why, well, I think in the past I have tried to do an experiment with very high salt concentrations, because here I only show you up to a variation of like going from a 0 millimolar to 1 millimolar. But I think in the past I have also tried to go a lot further, and the answer is yes, the particles stick. And I think this happens in general, because if I think of the literature, I don't think people have ever reported experiments going beyond this 1 millimolar concentration. I guess the reason is because the particles are just stuck, and or or in a sense, maybe they sort of become Brownian-like in the sense that they don't have this active component anymore. So that's a good point.
Yeah, I think 1 milliliter, 1 millimolar is quite high. Maybe, yeah, it's really quite high, 1 millimolar, so it can happen. Maybe it's also because of the double layer or the D balance changed, right?
Yeah.
Okay, yeah, next discussion. Thank you.
Thank you.
So any more questions? If no more question, we would like to thank Stefania again for his wonderful and nice talk and this wonderful
Thank you very much
discussion.
Thank you very much for inviting me. It's been a great pleasure. Thank you for all the questions.
Thank you for being part of this, and thank you all audience to participate in this mini talk.
Yeah.
Thank you.
Thank you.
I will stop
Bye.
sharing the screen now. Bye.